So, hi everybody and welcome to the SCAR Open Science Conference 2022, and especially to the session Environmental Factors, Diversity and Composition of Fossil and Living Antarctic Communities. I am Fabiana Canini from the University of Tuscia in Italy, and with my colleague Fernanda Quaglio from Universidade Federal de São Paulo in Brazil, we are commenting this session with the contribution of Dr. María Eugenia Raffi from the Centro Austral de Investigación Científica in Argentina as co-convenor. Before we start, just a few technical information. Each speaker will have 12 minutes and we have 8 presentations today. The question and answer session will be after 4 presentations. So you can write your question also during the presentation and we will read it after. We want to remind you that the organizer sent you a message informing the speakers that the presentation will be recorded and they will be available 24 hours within the session in the SCAR platform and in the YouTube channel. But if you want your presentation not to be published, please let us know and we will post the recording during your presentation. I really hope you will enjoy this session, the presentation and the discussion. Now I give the floor to my colleague Fernanda to introduce the speakers.
Okay, hello, good afternoon, good night, good morning for everyone. Thank you for all of you to be here with us. And the first work we have is by Carolina Costa Hospitaleche, Definitive Evidence of Terror Birds in the Eocene of West Antarctica. Please, Carolina. You are muted, so unmute your microphone. Okay, perfect.
Thanks. Well, I think the presentation is on the screen. Is that right?
Yes, it is.
Okay, thanks. Well, I'm going to present now a research I'm doing together with Javier Gelfo, Daniel Garcia Lopez, Facundo Irazoki, and Marcelo Reguero. The present research was motivated by the recent finding of a new fossil. It is a talon of a large bird of prey that was found during the last austral summer in an Antarctic field trip to Seymour Island. The fossil was collected from Ypresian sediment of the Coculea-1 allomember in La Meseta Formation in the fossiliferous site known as IAA213. This site is located in Seymour Island, Seymour-Marambio Island, due to the presence of the Marambio station in the same island that is part of the Shane Ross Basin. This is a small ice-free island which is located near the tip of the Antarctic Peninsula in West Antarctica, precisely in the northeastern side of the peninsula. This is the fossil. The fossil is almost complete and consists in a talon. It means an ungual phalange of a claw of a Jurassic bird. We can see here different features of the phalanx. Only the small piece of the tip is missing and is almost complete. For example, we can see here the curvature of the dorsal and the ventral surface of the phalanx, and a neurovascular sulcus running on both fascias, the lateral and the medial fascial. We can see in this picture that this sulcus is deeply excavated in the medial fascias than in the lateral fascias, and both tubercles, the dorsal, which is the extensor tubercle, and the flexor tubercle are rounded, and they do not project beyond the corpus of the phalanx. If we move the position of the phalanx, we can see it in a proximal view, like in the last picture, in the last photograph. And in the proximal view, we can see the general outline of the articular facet, and which is divided, asymmetrically divided in an articular medial cotilla in an articular lateral cotilla. All these features allow the assignment of this claw to the order Cariamiformes. Cariamiformes is a large group of birds, primarily flightless birds, which are represented in a fossil record from the late Eocene. We have also some questionable Paleocene records, but it is certainly recorded from the late Eocene to the late Pleistocene. And the family Cariamidae, which is represented today by the Seriemas, with two different genera. In the past, these birds exhibit a greater variety of forms that goes from 1 to 10 meters tall, and this is the reason because they are known in the literature as terror bird, because some giant forms are widely expanded in the fossil records. In general terms, they exhibit some features in common, like for example, the reduction of the wings, long and strong legs, and particularly strong and curved ungual phalanxes. Regarding the skull, they have large and long skulls, laterally compressed, and the tip of the beak is ventrally curved. All these features point to adaptation for running, a carnivorous diet, and also hunting skills. This group, the Phorusrhacidae, the terror birds, have been previously reported for this Shane-Ross Basin, but in all the cases, the records were dismissed. For example, we have here the first case, which is a left femur coming from Maastrichtian levels of the Cretaceous of the Vega Island that was recently reassigned to the Anseriform vegavis. The second case corresponds to a right tarsometatarsus that come from levels equivalent in age to the level where the fossil here described was found. It means it comes from the Prussian level of the Cuculea allomember in the La Meseta formation, and was reassigned recently to the Pelagornithidae of pseudotooth birds. Another case, the third one, is a small fossil, is just the tip of the bill that was reanalyzed, restudied, and assigned probably to a flightless rotite. It meets a ray form or some other Paleognathus birds. And finally, from the same levels that the last one, two unnumbered materials with clear morphological affinities to penguins that come from the uppermost levels of the Bartonial to Ritella allomember of the Submeseta formation. These remains were reassigned to Ephenicidae, which are a group clearly dominant in the sediments of the area. We have here a comparison of our fossil. Our fossils certainly can be assigned to a Phorusrhacid. We have no doubts about it. And we have here a comparative draws of the different genera of fossil Phorusrhacid and also from living Cariamidae. If we take account the curvature degree of the different surfaces, the dorsal and the ventral surface in the different genera of the Phorusrhacidae and the Cariamidae, plus the outline of the articular facet, the complete outline of the proximal in proximal view, and the morphology of the lateral and medial facet, we can assign this fossil to the second digit of the left hind limb. We have here two different pictures. At the left, we can see the position of this claw in a complete leg, in a complete feet. And at the right, we have three different draw of Cariamiformes. At the bottom, we have the living representative Cariama cristata, where we can see the position of the claw, Cariama cristata, the living representant, together with Chunga, the other genera of Cariamidae, have the strongest curvature in the claw, but we have here also Phorusrhacus longissimus, which is a large size Phorusrhacidae, and Philopteros colsecus, a small sized Phorusrhacidae, showing the position of the same element in the digit. the fossil here described fit in morphology and insight also to the large forms close to Photobrachus longissimus. So starting from that, we can confirm the presence of the Photobrachus in Antarctica. This would be the first fossil described with certain affinities with Photobrachidae. This is interpreted as a top predator. This form shares with flightless ratite, which are also recorded in Seymour Island, the large size and the inability to fly. But it also shares with falcons the curved talons and the predatory skills. Falcons are also recorded in Seymour Island. So with all this information, we can say that the specimen here described belongs to a terrestrial hunting bird, a giant carnivorous form that fulfill the role of top predator, apparently not occupied by land mammals in the Paleogene Antarctic communities. Photobrachidae like this one represents a gill hitherto unknown to Antarctica because they predate on large prey, and this way, with this new fossil, the continental Paleogene ecosystem and acquired a community structure completely different from those known in other regions of the planet, including South America and Patagonia particularly, where these birds are particularly abundant. Well, thanks for your attention.
Okay. Thank you, Carolina. We still have some minutes, so
I don't know if you have
no,
any
I think
questions.
it's better to wait until we will have a block for questions that will start at 3:51, but I would like to wait until we will have on time to present the next work.
Okay.
So we will be on schedule.
Okay.
And while that, people can include the questions in the chat, so we will make them later.
So I can comment also that this finding is very important for the area because it constitutes the larger predator in the area, talking about continental predators. We have other predators like the falcons I mentioned, but mammals, for example, do not have any representative like a large predator. They are all herbivorous in the area. Even if we compare with the South American fauna, it is completely different, the structure of the community. Oh, I saw the question about the ignites of the Kingshore Island. Yes, the problem with that fossil is that
Carolina, I'm sorry, we will have time for that. We just cannot
okay.
Sorry, I'm sorry, I don't want to.
Okay, I will continue later. Okay.
Cut you talk. Yes, because we will have a time for the questions.
Yes.
So, and now we are on the time to present the second work. I would like to ask the IT team to put the video. I'm sorry if I don't pronunciate correctly the name, but it's from by Betil Soilu, Diatom Stratigraphy and Biogeochemical Changes of a Periglacial Lake on Robert Island, Antarctic Peninsula. Please, when you're ready, put the video for us. Thank you.
Hello, I am Betül Bitir Soylu. I am from Institute of Marine Sciences in Middle East Technical University in Turkey. And here you can see my colleagues. Today I am going to talk about a part of our project which is called Diatom Stratigraphy and Biogeochemical Changes of a Periglacial Lake on Robert Island in Antarctic Peninsula. Polar regions are sensitive ecosystem, as you know, and Antarctica is the center of climate change observations. And long-term data is crucial for this research, but generally we don't have. At that point, paleoecology is an important tool to understand past environmental changes. And you can see here cores from Antarctica. These cores can be used as sedimentary records. And in these records, you can find also diatom fossils. And these organisms are used as key species. They have low tolerance for different environmental changes. So they can be used as biological indicators, and their another unique characteristics, their fossil structure are preserved well in the sediment, so it can be used for different environmental change understanding. And you can see here a diatom species from Antarctica, Planetodium australe. And here we try to understand changing ecosystem of the fragile Antarctic Peninsula and to explain the role of the climate and human impact in the ecological history in the Antarctic Peninsula, Robert Island. And you can see here the location of Robert Island, and here is the lakes that are sampled. As I mentioned, our sampling sites in Robert Island and Elvan Lake course used for paleoecology project, and our core length is 20 centimeter and it's taken from 1.6 meter. And you can see here Elvan Lake, its surface area is 11,139 meters square, and its surroundings is 470 meters, and its depth 1.5 and 6 meters, and altitude 30 meters, and it is distance from the coast approximately 500 meters. And different methods are used in this project. First of all, dating methods. It is used to have knowledge about the chronology of lake and sedimentation rates. And second one, loss on ignition analysis. This gives us information about carbonate and organic content of the core. And third one is XRF core scanning. Different elemental variations are observed by this analysis, and they could be indicators for different environmental changes. And another one is pigment analysis. This gives us information about the changing phytoplankton communities. And the last one is diatom analysis. Diatoms are used as key species, as I mentioned, by this analysis. So here you can see our dating analysis led to 10 and cesium 137. And in the first 10 centimeter, it goes back to 109 years ago, and its sedimentation rates changing from 0.66 to 0.12 per year. And here you can see loss on ignition analysis from S2 core, water content and organic matter actually values. And you can see upper part of the core, it is increasing through the upper part. So it makes sense because it's near to the today. And one point near the ninth layer, it's again changing. It's again, there's an increase. So here you can see the XRF analysis from S2 core. 27 different elements are observed in this analysis, and here I pick some of them. And calcium to titanium ratio, this is indicator for biogenic calcite production. And zirconium to rubidium, it's indicator for particle size. And iron to manganese for changing redox condition. You can see also in the other elements, it shows stable mostly, so we don't expect a drastic change in recent history of the lake. Here you can see pigment analysis in the core. We have different pigments here, you can see here, and it is composition, actually not absolute. It's percentage, not absolute concentration. And here are the groups for different pigment groups. And you can see after 1990s, there's an increase in some pigments, and which are mostly related to benthic algae. And also you can see here chlorophyll A has an increase by time, and this could be related to higher primary production by time, but also it can be related to degradation. So these results are very new. We want to add some other analysis to look at chlorophyll A and chlorophyll A derivatives to understand it is related to degradation or it's related to higher primary production. If it is related to primary production, this could be related to climatic conditions in the Antarctic Peninsula. And here you can see diatom analysis. There are three groups, as you see: green for neutral conditions, blue for alkaline conditions, and red is for acidic conditions. And they show different patterns through the core. And if we compare with the literature, you can see here the Navicula gregora. It is red star. It is higher in the earlier time of the lake. So it's mostly found in acidic lakes, and it is decreasing by time. And Nitzschia, you can see here different Nitzschia species. It is found in slightly alkaline conditions, and it is increasing towards the upper layers of the lake. But if you look at here, these are the most common species found in the core. They are related to natural conditions. So it's suggesting absence of drastic pH change, and range has been closely around circumnatural, we can say. And also, according to pigment analysis, earlier history in the lake, there is low primary production. After 1990s, higher primary production could be happened, and not a drastic change in biogeochemical properties are observed in recent history of the lake. So to sum up, sedimentary records give information about past events, and diatom analysis shows different groups were present in S2 core in Elvan Lake in Robert Island, and relatively stable biogeochemical properties are observed in the lake core, and first results give clues for different environmental conditions observed in Elvan Lake in Robert Island in Antarctic Peninsula. So our project is supported by Presidency of the Republic of Turkey and the Ministry of Industry and Technology and the Scientific and Technological Research Council of Turkey. Also, we have partners in this project: Istanbul Technical University Polar Research Center, Eurasya Institute of Earth Sciences, and Dumlupınar University. We thank to all supporters and partners. And thank you for your listening. Here, our team, Aquatic Ecology team, you can contact for your questions and applications.
Okay. Thank you for playing the video. We just wait 1 minute. I would like to ask the IT team to help us to admit Betil in the as a panelist. We are not able to do that, so we would ask you to do that for us.
I think she's not connected anymore.
Yes, but once she get inside the room, please help us to admit her.
Yes, ma'am.
Thank you. Okay. Now it's a turn to Javier Gelfo. Additions to the knowledge of the Astrapoteria and Lithopterna mammals from the Eocene of West Antarctica. Please, Javier.
Okay. Can you see the picture?
Yes.
Well, we are going to present here preliminary result of the latest paleontological fieldwork carried out in Marambio or Seymour Island during the austral summer of 2021 and 2022, particularly regarding the fossil record of land mammals. To begin, maybe we should understand the reason why fossil land mammals on the Antarctic continent are or should be so important. At present, there are no terrestrial vertebrates on the Antarctic continent. Birds and mammals are especially marine. When we say marine, we mean that they spend most of their life on the sea and they feed in this environment. They also present specific adaptations to their life in the sea, as we can see of the mammals found today, which are only represented by cetacean and pinnipeds. Without neglecting the historical reasons for the absence of living land mammals in Antarctica, it's important to emphasize that the terrestrial Antarctic biome is limited to 2 angiosperms and it's mainly composed of lichens, mosses, and fungi. Therefore, the primary productivity of terrestrial ecosystems seems to be very limited to maintain a community of terrestrial mammals. This generates an important contrast with the Arctic, the most comparable region for its climatic condition and its latitudinal location. In the Arctic, the tundra allows the development of a large community of land mammals, some of which have a wide variety of subspecies, like the Arctic hare or the reindeer or caribou. So fossil land mammals from Antarctica provide us with evidence of a history very different from the present. In the past, during the Eocene, communities of land mammals developed within the framework of terrestrial ecosystem, which were very similar in taxa composition to those of the southern South America in Patagonia. The fossil record in Antarctica is composed mostly by isolated teeth, which are harder than bones, and a few postcranial elements and footprints. And by now, they indicate that all land mammals belong to extinct species, represented by Tyrian mammals, like marsupial and placental mammals, and some Gondwanan Mesozoic lineage, which survived the Cretaceous-Paleogene boundary, like Dryolestida and Gondwanatheria. In the graph on the right, you can see the inferences of mass expressed in kilograms for the known terrestrial mammals in relation to the inferred feeding habits. To date, the fossil record indicates the presence of marsupial and Mesozoic lineage weighing less than 1 kilogram with insectivorous, frugivorous, and folivorous to granivorous habits. Plus that, there are placentals between 10 and 400 kilograms represented by Xenarthran and native South American ungulates orders. The last one are known by the Astrapoteria and Tartodon sobrali, and the Litopternas Parnoteriodontide, Notiolophos archinotiensis, and Notiolophos reyeroi. As you can see in the lower part of the graph, we don't have carnivorous, omnivorous, or strictly grazing mammals represented here. This could be a taphonomic bias. So, the three teeth and the scapula that we are presented here were collected on Marambio or Seymour Island. In this map, modified from Montes et al. To 2013, the Cretaceous Lopez de Bertodano Formation can be seen in green, which includes the Cretaceous-Paleogene boundary. The levels of the Paleogene are the Sobral Formation in dark orange, Cross Valley in violet, and for the interest of our talk, the Eocene levels of La Meseta Formation in yellow and Submeseta Formation in orange. On the right, we see the stratigraphic column with the detail of the allomembers of La Meseta Formation. And the fourth specimen discussed here came from the Cuculea 1 level, which is undoubtedly the most productive in this type of remains. The first specimen is the one on the right and belongs to the Astrapoterid lower right molar. It comes from the basal coquina level at DPB 284 and is clearly similar to the holotype of the only described Astrapoteria from Antarctica, Antarctodon sobrali, which you can see on the left. Both are lower right molars with the trigonid higher than the talonid and open lingually. But the new specimen, despite being broken in the talonid, is larger and more robust. Some other differences are due to the absence of wear in the metaconid and its round base, the protolophid wear facet distally projected, paracristid short but with, paraconid well integrated to the paracristid, and cristid oblique, strong and almost contacting the protoconid. These differences are probably due to a lossy variation and adds new information for the group. This is the second specimen recovered, which came from IAA 190. It's an upper, first, or second molar of a Sparnoteriodontid litopterm. In the occlusal view on the right, it's evident that part of the ectoloph is broken, as well as the distal part of the tooth, but the main characters of the Sparnoteriodontid are clearly recognized, for example, a bunoid and strong protocon, and the presence of an extra cingulum, which is more evident in the mesiolingual view on the left, where you can see the double anterior cingulum. The tooth is similar to Notiolophos archinotiensis, one of the two of the largest described Sparnoteriodontid from the area. The main differences with the new specimen rests in the large size of the hypocon, which is reduced in the M1 and 2, and very weak or absent in the M3 of Notiolophos archinotiensis, as you can see on the left. but particularly the new specimen differs in the absence of a low-fit metaconul, mesially extended and connected directly to the poorly developed hypocon. These differences seem to indicate the presence of a new ungulate taxa for Antarctica. The third specimen came from a small hill, IAA 213. The specimen is here interpreted as part of a low-fit of an upper tooth, but preserving only the external face with a basal cingulum, so it's very difficult to determine their taxonomic position. Despite this, it's belonging to a large mammal, probably a Sparnoteriodontid or a Strapoterid, since these are the only groups in Antarctica with vertical decussating Hunter-Schreger enamel bands. The enamels also exhibit dental wear facets, which are not very commonly preserved in the enamel found in Antarctica. So probably a mesowear and microwear analysis will give us good proxies for inferring feeding ecology among these groups. The last specimen that we present corresponds to a fragment of a scapula, probably of a large land mammal. It came from the middle section of Locutorio Hill. The scapula was found broken into several fragments, so the preliminary reconstruction of it can be seen here in different views, but the preparation is not already complete. The main recognizable characters are an ovoid glenoid fossa that don't have a glenoid knock, but as you can see in the right picture, present a strong supraglenoid tuber. The neck is very wide and thick, and the spine is partially bent toward the infraspinous fossa. Considering that the whole scapula is about 15 centimeters long, it is very likely that it belongs to one of the ungulates recorded in the area. Finally, and to summarize, we can say that land mammals diversity for the Eocene in West Antarctica continues to increase. The Sparnoteriodontid upper molars here described seems to indicate a substantial variation from the dental pattern of Notiolophos and possibly deserve the description of a new taxa. The other identified tooth give us new information to understand the relationship of the basal Astrapoterid from Antarctica and South America. And we have a lot of information to integrate from enamel and postcranial fossils. Larger mammals, mainly Astrapoteria and Sparnoteriodontidae, show clear adaptations in the enamel to withstand vegetable hard foods, but there is no development of ipsodont teeth. So the mesowear and microwear analysis will open a new window for inferences about the paleoecology of terrestrial ecosystems. Thank you very much for your attention.
Thank you, Javier. We still have a minute before the next presentation. So if you want to say something very short,
you can download here from the QR figure you have there some more information about Antarctic fossil mammals. I will be waiting if you have any question.
Okay, thank you. Thank you. So we'll go to the next
Should
work.
I stop to share this screen?
Yes. Cool, thank you. And the next work is by Carolina Perez, but it will be presented by Tamara Contador. And the title is, what do the wings of an Antarctic dipteran tell us? Deciphering wing variability in Paroculus steineni. Okay, Tamara.
Hi, thank you for everything. My name is Tamara Contador, and yesterday I'm presenting on behalf of Carolina Perez. I am, she's my student in my lab, so she couldn't be here today. So I will be presenting the work that we have been conducting on Paroclus teineni, which is a winged midge from Antarctica. Can you hear me okay? Because I am in southern Chile with very bad internet connection.
Yes,
Can you
we
hear?
can hear you very well. Yes.
Okay, thank you.
Yes, it's okay.
Okay, thank you. Well, so the presentation.
Sorry if I stop you.
Mhm.
Could you use the full screen mode for the presentation?
Okay. I will. Something happened. Hold on, I will do this right now. I was in full mode, but I don't know what happened. Okay. Can you see it in full mode now?
Yes, we can see it.
Yes.
It's fine.
Yes.
Okay.
Fine.
Okay. So our talk is about a work that we have been working on with Paroclus teineni, which is a native dipteran from the Antarctic and South America. So I will talk to you a little bit about the geographic scope of the work that we have been conducting, also the context of climate change and insects, and we'll present some of our results and points of discussion. So just to give you a little bit of context, the Antarctic today is composed of less than 550 species of non-marine invertebrates. And as we all know, the Antarctic is covered most of it in ice, so about 99.5% is covered in ice, but the areas that are free of ice or ice-free have a very diverse environment of lakes and freshwater streams that are active during the austral summer. And within these ecosystems, we find two insect species. One of them is the non-native and endemic Antarctic midge, Belgica antarctica, which is here in this photograph. It inhabits the Antarctic Peninsula and the maritime Antarctic and is freeze tolerant, is tolerant to freezing. And the other one is Paroclus teineni, which is winged, different from the non-winged Belgica. And this one is native to the Antarctic. It also inhabits South America, and it's only found in the maritime Antarctic or the South Shetland Island, and it's not freeze tolerant. So this is the habitat in which we find Paroclus teineni. These are the permanent deep lakes within the maritime Antarctic. I'm going to show you a series of pictures so you get to know this little insect. Here we have a photograph of the female and the male. The male has puffy antenna like you see here, or plumose antenna. This is the larva. It's considered an aquatic insect because it has an aquatic phase, which is longer than the terrestrial phase, the adult. The pupa is also aquatic, as you see here in this photograph. The adult emerges out of the water, and they are very abundant in the edge of the lakes in the maritime Antarctic, like you can see in this photograph here. So in this context, we have become interested in studying Paroclus teineni as a sentinel of climate change in Antarctic and subantarctic ecosystems, given that insects are highly affected by climate change today. We have seen that there is dramatic rates of decline in the world's insect species, and climate change is affecting their phenology, their distribution, and behavior at all trophic levels. Also, we know that polar regions are greatly affected by climate change, and that over the last decades, King George Island has experienced the loss of glaciers, and at the same time, because of this, new ice-free areas have appeared. Therefore, we have studied the distribution of Paroclus teineni through the whole maritime Antarctic. And we have also, in our previous studies and ongoing studies, have developed distribution models that have showed us that in projections with the different climate change scenarios, we could see an expansion of the distribution of Paroclus teineni from the maritime Antarctic into the peninsula. We have also studied the ecophysiology and life history of this midge in the Antarctic, and we have seen, as other studies have also seen, that Paroculus steineni is not freeze tolerant, but it has a wide thermal tolerance range. So it can live in cold environments that do not freeze, but also it can stand higher temperatures. Therefore, it could expand its distribution. And that the life cycle within the Antarctic lakes, it's very long. It could be as long as 4 years. And so we are still studying this, but we think it could be up to 4 years given the temperature accumulation in the lakes over this time period and the observations that we have done both in the field and in the lab. So in particular today, we are going to focus on one of the studies that we're conducting on Paroculus steineni that have to do with adaptation and phenotypic plasticity. In particular, we want to see how wing morphology may be an indicator of environmental stress in the context of ongoing change and the stressful environment in which the species is found. So we have, given the geographical heterogeneity and extreme environmental conditions in the maritime Antarctic, we hypothesized that wing morphology will be significantly different between populations of Paroculus steineni within and between islands. So what we do here, well, we focus particularly in populations found in King George Island, and we sampled 10 lakes in King George Island and 3 lakes in Nelson Island. We sampled a total of 366 individuals, of which about 250 were females and 100 something were males. And we conducted a geographic, a geometric morphometric analysis of the wing, identifying 13 specific landmarks, as you can see in this photograph. And we later analyzed this with Morpho-J to see how the differences in wing morphology and shape could be identified, if any. And what did we find? So we found that there is, of course, we knew that there was sexual dimorphism between these species, but we found that the difference in shape and wing size is evident, but not on wing size. So we see a difference in shape between the male and the female, but not on the size, as you can see in these 2 distinct groups. Also, we found that there's differences in wing shape between populations in the different lakes, and also between female and males. So we can see that there's 4 distinct groups in this very small geographic area, and in particular in female populations. So if you can see here in this map, we have the lakes that we sampled, but we also have 4 distinct groups of distinct wing variation in female populations. So just to give you a quick conclusions, using the geometric morphometric approach, we see significant differences in the wing shape and between male and female individuals, particularly from Gluboko, Kittich lakes, and also in other lakes, but in less proportion. And we are also conducting molecular analysis to see if we actually have distinct populations of this midge in this small geographical area. And we have found using microsatellites that we have distinct populations, but some populations do not necessarily correlate with the difference in wing size and shape. So we are seeing both phenotypic plasticity, but at the same time, distinct structure within the communities of Paroculus steineni. So what are some of points of discussions that this brings us? Well, we know that Paroculus steineni has a wide thermal tolerance range, but we wonder what other factors could influence its possibility of expansion in the maritime Antarctic, given that our models indicate that it can expand. So when we study the population structure at this small geographic scale, we see that there is some connectivity between the populations, but not complete connectivity within a small geographic scale. So it may not be completely able to move, but also we have to study the dispersal mechanism. So is it dispersed naturally by wind, or is it dispersed by the help of birds, for example? So these are some questions that we are asking right now to see if the expansion of the population of Paraculus tainani could actually occur or not, or if it could become in danger in the maritime Antarctic given its limited dispersal capabilities. So this is what we were showing today. Many thanks for listening, and thank you for allowing me to present in terms on behalf of Carolina Perez.
Thank you, Tamara. We still have a minute before our question and answers time. So if you want to talk, or
if someone has a question, you can write it down in the question and answer box.
Okay. Okay, if someone has a question for one of the presenters, just write it up. Or if you can open the microphone because it's easier than write, it's okay.
I think the participants cannot open their microphones, so we have to allow them. So, but if you want to write us, we can allow you to open your microphone if you want, if you prefer.
Yes. Okay, I have a question. If nobody has a question, I have a question. It's not a specific question because I don't work with mammals, but it's for Javier. About the ungulates, is there a difference of preservation quality between material from Antarctica and South America? There's a big difference because I think it can be related to how trustful is the fossil record if you have more quality or bigger, a higher number of bones preserved. I think it can affect how you can study in both regions. So.
Well, no, there is not a preservation difference between southern South America, Patagonia particularly, and Marambio. But the main difference is that all the stratigraphic sequence in Marambio or Seymour Island is marine. So all the land mammals we found there are transported and came from around 10 kilometers where the coast was present. So we found a lot of terrestrial fossils like plants, woods, flowers, seeds. So our record is clearly biased by the transportation we have. And this is the main difference with Patagonia. In Patagonia, we have all terrestrial sequence in the, for example, the Rio Chico group in Patagonia, which cover most of the Paleocene and Eocene, the Sarmiento Formation. But here our only record of mammals, despite some footprints assigned to mammals in the Shetland Island, all the record came from Seymour Island. So we are very constrained in order to identify correctly the diversity of mammals in the past. We are only looking from a a small window to the past. We need to find some terrestrial stratigraphic sequence with another kind of preservation.
Okay, thank you.
You're welcome.
No more questions?
I would like to
Okay.
comment on the question about the ignites, the footprints of Kingshore
please.
Island.
Please.
Well, the assignment of this footprint to ratite or for racus was due to the previous reports of these birds in the Seymour Island, not based in the morphology of the footprints. So these footprints are not informative in systematic terms, and they could be assigned either to this group or other group of birds. So they need to be revised, and probably with new findings, we could have a more clear scenario. But the main feature to assign them was the previous report in Seymour Island that were later dismissed.
Okay, thank you.
You're welcome.
Okay.
I have a question for Tamara, if there is enough time.
Yes, we still have time.
Okay, Tamara, hi. Do you have
Hi.
any idea how similar the wing patterns are between the chironomids and the particular Paroclus steineni and the mosquitoes? Is it possible to confuse them?
Tamara, can you open your microphone to
I'm sorry,
answer
the
question?
internet fell, but now it's back.
Oh, okay.
I
Okay.
couldn't hear the question.
Okay, I wonder if you know how similar are the wing patterns between the chironomids and, for example, a mosquito. Is it possible to confuse them?
No, they are very different, and it depends on the
Okay.
species. They may have the wing venation is particular for the species,
Okay.
and so they are very distinct for each species. So
Okay.
in this particular, the wings that we obtained from fresh adults that we collected, but it would be interesting to see if there's any sediments that we could collect from the deep parts of the lakes to see if there's any fossil records. I know
Okay.
that Paroclus steineni has been collected from paleolimnological samples, but the head capsule. So, but I haven't seen studies that show, for example, if there is any wing. Of course, the wings are going to be harder to find, but there is some records of head capsules that indicate that Paroclus steineni has been an Antarctic inhabitant for a long time.
Okay.
Mm-hmm.
I will then contact you by email.
Yeah, perfect.
Thank you.
Mm-hmm.
Thank you. Cool. We still have time for questions. If nobody has, I have a question. I always have a question. So, and this is for Tamara. Have you a comparison between your data from Antarctica to the Arctic? Is there a big difference? There are some representatives in the north.
Yes, in fact, there is actually there is some studies that have been done on Arctic deep terrain, particularly chironomids, and the life cycles are also very long. Some of them in the Arctic are up to 7 or 8 years. And we have seen that here we think that it's about 4 years, the life cycle of Paroclus steineni. Also, the genus Paroclus is found in the poles. So I'm also looking for information if there's any research on Paleoclus from the Northern Hemisphere, but other chironomids behave similarly in the Arctic regions.
Cool. Thank you, Tamara. So now we are on time to the next presentation. This by Tamires Morila de Souza, Paleoecology of the bivalve mollusks from La Meseta Formation, Eocene, Seymour Island, Antarctica. Tamires, you can present when you're ready.
Good afternoon, everyone. My name is Tamires. I'm going to present my work named Paleoecology of the bivalve mollusks of the La Meseta Formation, Eocene, Seymour Island, Antarctica. So the most glaciation episode in Antarctica is related to the greenhouse and icehouse changes that occurred in the Cenozoic, early Oligocene, which is materialized by the formation of Antarctic ice sheet. To understand what happened during this period, we have the La Meseta Formation, which exhibits a cooling trend along the Eocene, with milder temperatures prior to the formation of ice sheet in Antarctica. This location contains bivalve mollusk fossils that are a great group to study climate change and fishes because their life habits are directly associated with the substrate. And to understand how biota have responded to these changes is crucial to predicting how climate change affected different biological groups. Here in the left side, we have a geologic map on the La Meseta. In this yellow part, represents the location. My work aimed at understand the evolution of the guild as ecological parameter in parallel with the sea deepening as well as sedimentary filling of the unit. Then we use a descriptive statistical analysis of the bivalve species and guilds as an ecological parameter in parallel with the sea deepening as well as sedimentary filling of the unit. So here, the table that I made in my work, and here we have the genders, here the species, and here the terms, which means tertiary Eocene La Meseta. These terms, the number is 1 to 7, and when we separated the data from Stewart-Meister in a binary character from 0 to absent when these species weren't present, and 1 when they were present. And this other part of here, we have the guild. So this guild was named for Abraham in 1994. Then we made this other table. Here we put the guild number G1 to G16, and we made a sigla for this name using the first letter of the same word here. And after that, we made this translation for Portuguese because these terms were not very common in Portuguese. And I used then that information to make my conclusion of graduation studies. My results, so we made this graph, which shows the distribution of guilds by the stratigraphy of the La Meseta Formation. And here, the letter N represents the total number and the species described for each term here. And the bars shade of green, for example, represents the infaunal animals. In blue, the deep infauna animals. Orange, shallow infauna. Purple, semi-infauna. In red, the boring. And yellow, epifauna. So my analysis indicated that the basis of the La Meseta formation here in the term 1 and 2, where the facies granulation scores, both guilds and species here are less diverse. For example, here we have G5, facultatively mobile infaunal deposit feeder, G4, facultatively mobile deep infaunal cremosymbiotic, G8, facultatively mobile epifaunal suspension feed. All these guilds here represent a number of 7 animals in this term. So it's a low number for this part. But when we open the stratigraphy, here, for example, in term 5, we have other guilds, for example, semi-faunal animals, and this kind of guilds become more recurrent. In this term, we have a huge number of N, we call 36, and other guilds. For example, I put here G16, 15, facultatively mobile infaunal cremosymbiotic, and facultatively mobile infaunal suspension feeder cremosymbiotic. This huge number for the term 5 represents some then the water in this part of the stratigraphy, it's also I forgot the word. This is respond to the sea level rise. That's what I'm gonna talk to you. So here we have a low level and here an upper level of the sea. And what means the term 4, for example? Then we have a low number comparing to term 5 and the others are here. That can be related to the presence of a transgressive lag, and maybe it's a consequence of sedimentary filling in relation to change in facies and guilds as an ecological parameter. So my analysis explains the change of the guilds and the faunistic composition along the stratigraphy of the La Meseta formation. My analysis corroborated the physiological description of the formation made for Marensis in 1994-95, when he described the formation as an estuarine environment with river influence at the base, here at the base, and evolving to a deep water environment to the top of the stratigraphy. But this was the first analysis I made, and we need to analyze other paleoecological parameters. For example, bathymetry, temperature data, and biogeography distribution of the current representative, because we need to understand how such environment characteristics potentially influence the dynamics of the marine biota. So comparing the fossils with the now living. But we need to add to taxonomy mailing at the levels. For example, in term 4, when we have a transgressive lag, and we need to understand if we have some possible atomic mixtures. So here's my references, which I made to build this presentation. And I want to say thank you for all for listening. And it's that. Sorry for my speaking fast.
Okay, Tamiris, thank you for your presentation. We'll have time for questions in the next block of questions and answers. And we still have 2 minutes before the next presentation. So if you want to add some commentary or some information you want to talk, you can do it.
No, I just want to say I'm gonna study this other data net and other analysis in my master's, which I almost passed it. So we have more work to do now. It's that. Thanks.
Hello, this is Manoj from Tech Team. Do you want me to start the recording for Eric?
Okay, but you can just prepare the video, and we just wait 1 minute to start the video because we want to be on time. So just wait a little bit and prepare it. Thank you.
Yeah, okay, thank you.
Okay, now the next presentation is by Eric Du Châtelet, but it is presented by Maywin Herleden. Sorry for the pronunciation. And the title is Biodiversity and Paleobiodiversity of Distate Amoebae on Kerguelen Archipelago, a key for climate change monitoring. Please play the video.
Hello everyone, my name is Maïwen Herlédan. I'm currently in PhD at the University of Lille in France, and I'm supervised by Eric Armino du Châtelet and Viviane Bout-Roumazeil. And I'm working on a project of the French Polar Institute, the program Envicaire. And today I'm going to talk about the biodiversity and paleobiodiversity of testate amoebae on Kerguelen Islands, to see if it's okay for climate change monitoring. So first, I will introduce you the testate amoebae. There are single-celled microorganisms that are recovered by a shell. The shell can be organic, can be siliceous, or can be made by different minerals. And we can see here some illustration of different shape of testate amoebae. The pictures was taken by scanning electron microscope. So testate amoebae are living but essentially in wet environment like pit bog, river or lake. We can find them all around the world, and they are really good markers of environmental changes because they're fossilized in the soil. Kerguelen Islands are located at the south of Indian Ocean in the subantarctic region, near from the polar front. The weather of Kerguelen Islands is really, really extreme, with a lot of winds and a lot of precipitation. It is really interesting to study testate amoebae on Kerguelen Island because with lot of precipitation, we found very, very much T-state amoebae. And here we can see on the graph the rainfall trend and the evolution of temperature since 1950. And we can see that there is an evolution in temperature. So the average temperature has been rising since 1950, but there are no significant trends in rainfalls. But in the literature, they said that there is a decrease of precipitation since 1950. So there is an evolution in the climate on Kerguelen Island. So the problematics today are: what is the current diversity of T-state amoebae on Kerguelen Island? How does this diversity vary over the time? And what are the parameters that can influence this diversity? So to answer to these questions, first, we have made some samples on Kerguelen Island in 2019 and 2021. So we have made some recent samples, plant samples like terrestrial moss and Azorella selago. This is an endemic plant of Kerguelen. And also we have made some past sample with sediment cores. So after sampling, we have prepared and analyzed all the samples in a laboratory in Lille with some geological data to better understand the type of environment. And the last step of the analysis was to observe T-state amoebae under microscope during hours and hours to recognize the different species. So in this study, we have studied 85 samples of moss and Azorella selago, and a total of 124 species have been found. The samples have been made all around the archipelago with a different type of soil, like fell-filled, vegetated soil, or wetlands. So there is a different characteristic of environment. And this data can be a really good things to do an ecological affinities of some species. Like on this figure, we can see that some species have some affinities with the quantity of total organic carbon in the sample. So here we have some species that have an affinity with low total organic carbon, and some other species that have an affinity with high total organic carbon. So we have some difference of the species, and we have the same thing for the water content. So some species like a very low water content, and some other like very high quantity of water. So with this data, we can work on sediment cores and try to understand the evolution in the past, the evolution in environment in the past. So we have made two sediment cores in 2019, one in Valstuder at the northeast of Kerguelen, under an Azorella selago cushion. So the bottom was dated to 150 years before present, and we have made 17 samples. And the other one was taken in Presqu'île-Ronarc'h, as the southeast of Kerguelen, under Acena magellanica plant, and the base was dated at 2010 years before present. We have made 20 samples. So first, here you have the results of Suazo sediment cores. Here you have the percentage of each each T-state amoebae compared to the depth of the core just here. So we can, the top of the core is the most recent and the bottom is the most older. So we have three different areas. The first one at the bottom of the core was characterized by the absence of this species. The species was composed by an organic or siliceous shell. So, and we have also an abundance of these two species that are mineral shells. So it signifies that there is a low total organic carbon in this area, and we are in a mixed zone with low humidity. After, we have a second zone that it's an intermediate zone with total organic carbon medium, and we are in vegetated to mixed wetland. So this is the intermediate between the bottom area and the top area. And here we have a high total organic carbon, and we are in vegetated wetland with the high presence of siliceous and organic shells. If we are looking for the difference between the type of shells, we can see that organic and siliceous shells are more present at the top of the core than mineral shells are more present at the bottom of the core. So mineral shells are well preserved than the other shells, so maybe there is two bias of preservation. For the other cores, we have four different areas, four different zones, and we can see that the evolution of total organic carbon is constantly decrease along the core. So at the bottom, we have a high total organic carbon and vegetated wetland. Then at the top of the core, we have total organic carbon low to medium, and we are in a mixed zone with less vegetation. If we compare the two cores, we have some local effects in each core, and a global effect can be affect the evolution of environment. So in Studer, the core can be influenced by the growth of the vegetation, and the proximity of a river can affect the water content. And in phonolith, we have a drying effect along the core that can due to the variation in water level, because there is a river near from the core, and a difference in sedimentation rate. So also the freshwater flow of the river can influence the core. And also it can have a climate change effect with the increase of temperature and decrease of precipitation. And Damien Fourcy has also observed that there is a drying effect in other parts of Kerguelen, Iceland, so it can be really a global effect. So to conclude, recent database has shown 124 species, so it's really cool. We can say that T-state amoebae contribute to environmental study because we can find them in the different cores. We can see difference between the two cores that can be due to local effect or global effect. So that is also another question. And we can see also some difference in composition of shells along the core, maybe due to the preservation of the shells, or maybe due to the concentration of minerals in the soil. And to complete and compare this data, we can study other cores spread over the archipelago and better understand the evolution of environment. So thank you for your attention. And if you have some question, I can answer them. Thank you.
Okay, thank you. May Wen, I hope I'm pronouncing correctly your name. Thank you. The next presentation is by Graham Lobert, Effects of Temperature on Early Embryonic Development of 3 Antarctic Invertebrates. Graham.
Hello, everyone. Can everyone hear me all right?
Hello. Yes, perfect.
Perfect. So hello again, my name is Graham, and I am currently a PhD student at the University of Hawaii in the Moran Lab. And today I will be talking about some of the work we've done over the past few seasons at McMurdo Station on the effects of temperature on early embryonic cleavage of 3 Antarctic invertebrates. So as all of you know, the Southern Ocean is the ocean that surrounds the Antarctic continent, and it is isolated by the Antarctic circumpolar current, which keeps the waters around the Antarctic very cold. And from our temperature data loggers in McMurdo Sound area, the annual temperature varies by less than 3 degrees Celsius, and that's from benthic data loggers. And so 3 of the Antarctic ectotherms that I'll be talking about today, Tritonia labelli, a nudibranch, and 2 sea spiders, Amathea glacialis and Nymphon austral. You can see them pictured here, are stenothermal and have a very long development, with Tritonia labelli and Nymphon austral, the ones on the ends, having a development of roughly a year, and Amathea glacialis having a development of roughly 3 years. Under Tritonia labelli here, you can see an egg mass. That's this white tubular structure coming out of it. That's what we would collected. And underneath this sea spider here, you can see another egg mass, this orange underneath it. And while I just said that there's very long development, it would be very nice to be able to see them through some of their developmental stages. But today I'll be talking about early cleavage. And here's pictured 1 cell through 8 cell. And we measured them up to 32 cell for Tritonia labelli. We measured it through 32 cell for all the species, but here pictured is just Tritonia labelli. And the reason why we're working with early cleavage is because it's experimentally practical, not only because of those long developmental times, but it's very easy to classify these as distinct stages. So 1 cell, 2 cell, 4 cell, it's very easy to see that. Second, early cleavage is known to be a very temperature sensitive process, and coupling that with these stenothermal organisms, we want to see if that stenothermality transfers through to early development, if we see those same trends as stenothermality in early development. And then lastly, but probably the most important, is that early cleavage is vital for successful development. If these organisms aren't able to get through early development, nothing else really matters. You get what looks like down here in the corner where we have abnormal growth, and this organism ceased to develop past this point. So early cleavage is very important for these specific organisms to be able to grow into an adult and contribute to the population. So if they can't make it through that, nothing else really matters. But for our experimental approach today, we measured the effects of temperature on rate of early embryonic cleavage across the -2.2 to a positive 2.2 degrees Celsius. And the reason why we did this temperature range is because this is just a little bit above and a little bit below what these organisms would experience in the McMurdo Sound area. And to compare these, we used 2 metrics, the first being Q10, which is a metric that standardizes any given rate change over a 10 degrees Celsius. And in most biological processes, we see a Q10 of 2 to 3. That's based on the metabolic theory of ecology. The second metric is the Arrhenius analysis, and I'll get into that on the next slide, as it's a little bit more complicated to explain than just a verbal explanation. And then second, we wanted to test this over multiple egg masses within a species, and then between those 2 distantly related taxa, the sea spiders and the nudibranchs. And so here are some quick graphs I pulled off Wikipedia that help to explain Arrhenius transformations. I want to draw your attention to this graph on the left here, where we have temperature and the rate of a chemical reaction. And as you can see, as temperature increases, the rate of that chemical reaction increases as well. And when we transform this, we change the X and Y axis, and we get a more linear looking line. And from that, we can do a line of best fit. And with using the Boltzmann gas constant pictured down here, we can infer the Q10, the activation energy of the system, and then as well as Arrhenius breakpoints, which these breakpoints help us to infer a change in thermal sensitivity occurring in this system. And to give you some context of what that means more biologically and not necessarily just this chemical reaction, here is two graphs from Crapsey et al. 2021, in which they monitored the early development of a fly and a frog species. And you can see here that these blue points are the developmental rate that's best described by that line and that activation energy above it. Those red points are where it falls off. That's where there's a change in some of the underlying kinematics of early development, and it's not described by that activation energy above. And so these points are where there is a change in thermal sensitivity. And I want to draw your attention to these upper and lower limits. You can see at the upper and lower limit, there's a drop off, because we won't see the upper limit in some of the data I will be presenting you today. But for our experiment, we took an egg mass pictured here. You can see all the little embryos in there. We took uncleaved zygotes, placed them into a glass vial with filtered seawater. And then we placed the vials into an aluminum thermal gradient. And this aluminum thermal gradient had glycol loops coming in both sides here that kept it a nice temperature from minus 2.2 degrees Celsius to 2.2 degrees Celsius across this 12 rows of temperature. We placed 3 replicates from each egg mass in there. And then from that, we monitored it daily to stage them at 1 cell, 2 cell, all the way up to 32 cell. And then we got a developmental rate. And from that developmental rate, we make a graph that looks like this, with temperature on the x-axis, developmental rate and cleavages per hour on the y, with seasonal variability here in the middle to give you some context of where these organisms see throughout the whole year. And each of these points here is the developmental rate of a single vial, with there being 3 replicates at each. A lot of these overlap, so you'll only see 1 point. And to give you some context of what this cleavage rate and cleavages per hour means, at the lowest temperature at minus 2.2, it took 18 days to get to 32 cell, while at the highest temperature, it only took 7 days. And if we calculate the Q10 of that, we get a Q10 of 7.8, which is much higher than that 2 to 3 that I said we would expect for most biological processes. And then we transform that erroneously, and to help orient you with this graph, again, we have temperature on the x-axis, developmental rate on the y-axis, just transformed. And now the lower end of the temperature scale is down here on the right, and the higher end is on the left. And we do a line of best fit, and we can calculate the activation energies, the Q10s, from this, and we get a Q10 of 7.3. And while this line, or this plot is best described by one line, using broken stick analyses, most of the data is described by 2 lines, which means that there's a change in thermal sensitivity. There's a change right about at this point in their thermal sensitivity. So we calculated the Q10 before the breakpoint, we got a Q10 of 14.7, and then we calculated the Q10 after the breakpoint, and we got a Q10 of 6.9. So we can see those high Q10s before the breakpoint dropping down to a little bit lower, but still higher than what we'd expect to see, with the breakpoint right around minus 0.6 degrees. And the really interesting thing about this breakpoint is that it occurs within their natural temperature range. So within the temperatures that these organisms would see throughout a year, there is a change in their thermal sensitivity, and they are more thermally sensitive in their natural temperature range than they are outside of it. And then secondly, the interesting thing is that even though we got this up to upwards of 4 degrees above their ambient temperature, we do not see a drop off that we saw in those other graphs here. So maybe they're able to withstand some higher temperature. I'm not sure quite why we haven't seen a drop off yet. But to summarize, we had some that had a breakpoint, or we had, majority of them had a breakpoint, but we had some that without. And so 7 of the 10 egg masses of Tritonia labelli that we worked with had a breakpoint with an average breakpoint minus 0.6 degrees, right within their natural temperature range. And then those Q10s changed from around 36.6 to 5.1. So again, a very high Q10, very high thermal sensitivity before the breakpoint, and then dropping down to a lower thermal sensitivity after that breakpoint. And then this was even seen in the ones that without a detectable breakpoint, we had a very high Q10 throughout the whole graph. And so for some conclusions, there was a large effect of temperature on cleavage rate throughout the temperature range. So there was a large effect. These organisms were very thermally sensitive. That's indicated by those high Q10 values. And in 7 of the 10 egg masses, they showed there was a change in thermal sensitivity. I want to hit that point home that they were more thermally sensitive in their natural temperature range than outside of it. And these patterns are shared among distantly related taxa. I talked about the nudibranchs, but here are some of the sea spiders. Nymphon austral here on the left and Amathea glacialis here on the right. We saw those breakpoints for Amathea glacialis at -0.8 and Nymphon austral at -0.6, again, within that natural temperature range. And then the Q10 being very high. This is the first number I have listed here, 82.3 and 22.7 respectively, dropping down to 3.8 and 6.9, still over the board, higher than that 2 to 3 we would expect. But that is all I have for you today. Thank you to the National Science Foundation for funding this project and all the help we've got from the McMurdo community and the University of Hawaii community. Thank you all for coming out today. Be sure to stay around for the talk after this, as another one of our lab members will be talking about some of the work we've done. And then tomorrow at the same time, our PI will be talking about some of the work that we've been doing. If you have any questions, feel free to email me at gtloberts@hawaii.edu. Thank you.
Thank you, Graham. We still have a minute. So
I don't have anything else to add, but thank you all for coming. So.
Okay, thank you. So we wait just a little bit, 1 minute for the next presentation. Okay, then. Next work is by Ming Wei Aaron To. Sorry for the pronunciation.
No, you got it right.
Okay. A heartwarming snail using cardiovascular measurements to investigate the thermal acclimation potential of an Antarctic marine gastropod.
Thank you so much. Just want to check, can everyone hear me fine and see my screen? We're all good. All right. So,
yes,
hey, everyone.
Perfect.
Thank you. Thank you so much for staying around for my talk. My name is Aaron. I'm a graduate student from the University of Hawaii. And I'm really excited today to share some of the research that we've generated in our past field season at McMurdo Station. We just heard from colleague Graham about how some of the cleaving embryos of Antarctic invertebrates react to heat. And I'm going to pivot away from that early stage angle and talk about how we assess the ability of some adult snails to acclimate to warming waters by measuring their heart rate. And so the issue at the forefront of Antarctic marine science is, of course, climate change, with the forecast being that Southern Ocean sea surface temperatures are likely to rise between 0.5 to 1.7 degrees Celsius within the next century. Antarctic animals are notoriously stenothermal or temperature sensitive, so it's really important to understand how these animals are going to react to warming waters. And there are generally 4 main reactions that species have to warming environments. The first thing that they can do is they can move away. Okay, they can move to cooler places. However, migration is not really achievable for Antarctic animals as they exist at the physical limits of temperature, which is the freezing point of seawater. They could adapt, sorry, wrong way. They could adapt, or they could evolve traits that help them to deal with heat, such as these land snails over here, which are evolving lighter shells for thermal regulation. However, because as Graham mentioned earlier, rates of growth, lifespans, generation times, very long in Antarctic invertebrates and ectotherms, which is associated with slower rates of adaptation. So adaptation is not likely to be that mechanism that is going to save Antarctic species in the face of climate change. What is more likely to help is acclimation, okay, which is the individual animal's ability to change their phenotype, okay, from one stable state to another in response to some kind of a stressor. The poster child for acclimation is the tilapia, which is able to change its bodily functions to survive in both fresh and salt water. And finally, if oil fails, the species will go extinct, which is a non-ideal but unfortunately common reality for many species. But of course, we hope that it's not going to come to that. Okay. The importance of acclimation over adaptation in Antarctic ectotherms has been recognized and has been studied before, with a general consensus from studies being that Antarctic ectotherms are really poor acclimators. Some invertebrate species take up to 5 months to show any sign of thermal acclimation at all, which is really, really long, to the point that it doesn't make any ecological sense or purpose to the animal. Now, what do I mean by that? So over here, I have a graph of seasonal temperature variation at the benthos of one of our dive sites, Cape Evans, which is in McMurdo Sound, a very high latitude location. So our group took these data by leaving data loggers out on the benthos between November 2019 to October 2021. The data logger was positioned at about 90 feet, and it was constantly logging for about 2 years. So what we found is that we detected these like really warm pulses, which reach up to 1 degree Celsius, which could represent an increase in something like 2 degrees in as little as 2 weeks to a month. Okay, that 5-month acclimation period that we mentioned earlier would be ecologically irrelevant, as by the time these animals have finished their acclimation response to warm pulse, we would be kind of right back in winter. And so this brings us to our question of the day. Can we find evidence of thermal acclimation in Antarctic invertebrates on a time scale that's somehow consistent with seasonal and variation in environmental temperature? And to assess acclimation, of course, the first thing we need is an indicator. And we decided to use heart rate as a measurement, as an indicator of acclimation. The heart as an organ works essentially by sending oxygen-laden blood to the rest of the body in response to some kind of oxygen demand. And so it's often used a good indicator of how well an animal is performing. So a very brief diagram of our setup over here. We basically use an infrared sensor. It's kind of similar to the ones used in the hospital. We have to attach that to a shelled organism, and then it detects kind of faint changes in infrared light transmission as blood vessels throb, and then eventually logs that signal as a heartbeat. We chose this animal as our experimental organism, Neobuxinum eateni, which is a shelled Antarctic gastropod, fairly large snail, maybe about 3 inches long, has a circumpolar distribution and a readily detectable heartbeat, which is kind of perfect for our method. So what do we do next? After finding out that we want to use the snail, we went out diving. We went out to collect these snails at 4 sites in McMurdo Sound. So McMurdo Sound is located over here in Antarctica, and we went to these 4 sites that were kind of accessible from McMurdo Station. We have Turtle Rock, we have Cape Evans, we have New Harbor, and we have Granite Harbor as well. So here is what the benthos looks like at our 4 sites. It's typically kind of rocky, often with soft sediment. And we found these snails from anywhere between 20 to 130 feet, which is the limit of how deep we're allowed to dive. We put them in these like coolers, and so that they stay nice and frosty for the trip back to base. And then we put them into these flow-through sea tables that we have on station, which keeps them cool until we're ready to do an acclimation procedure on them. So what we did next is to stratify the snails according to location so that they have balanced representation in our treatments. And then we randomly assign these snails to one of our 2 temperature treatments. So first we have -1.8 Celsius, which simulates the freezing point of seawater ambient temperature that these snails were taken from. And then we have our elevated treatment, which is a 4 degrees Celsius, kind of warm. And so we put them in these 20-liter buckets for 2 weeks. And one of the snails actually laid eggs during our procedure, so they couldn't have been too unhappy with the accommodations. Again, we mentioned that we decided to expose these snails for about 2 weeks, which is kind of short. Remember that previous papers said that invertebrates take up to 5 months to acclimate. But again, we're trying to see if they could acclimate in a short time frame. And here is a picture of our setup. We put the snails in this little jacketed beaker and hook it up to a water bath, which controls a temperature ramp, which I'll talk about later. Okay, so that temperature ramp, see, here it is. We subjected them to a temperature ramp spanning -1.8 Celsius to 18 degrees Celsius. So that's about a 20-degree shift, 2 degrees stepwise increase in temperature happening every 2 hours. We constantly monitored their heart rate and water temperature. And so this is if you ever wanted to see a snail's heart rate, this is kind of what it looks like. This is a 50-second period, and each one of those peaks represents a heartbeat. Okay, so I then proceeded to manually count each snail's average heartbeat for each temperature, and we come up with a graph like this for each of our snails. So on the y-axis over here, we have heart rate in beats per minute, and on the bottom, we have temperature in Celsius. So the first thing you'll notice is that heart rate increases quite quickly with temperature up to a certain point, and then it starts to drop and starts to tank really, really quickly, which is very typical of a thermal performance curve. Okay, the animal is able to keep up with the stressor until a certain point, and then its body is no longer able to withstand it, and then it starts to give up. So what in this graph is going to let us assess acclimation? The first thing that we want to know is the maximum heart rate that each snail managed to achieve, which tells us the capacity of each animal to move this oxygenated blood around its body. And second, we're interested in the temperature at which that maximum heart rate was recorded, which I'll refer to as the peak temperature. Finally, we're interested in the heart rate breakpoint, which is where the snail's heartbeat can no longer keep up with the demands of its body and starts to fail. This breakpoint was calculated using an Arrhenius transformation, which Graham adequately explained just now. And then we proceeded to run t-tests on each of these 3 parameters, looking for differences between the -1.8 and 4 degrees Celsius exposed snails, with the expectation that better performance at higher temperatures can be taken as a sign of acclimation. All right, a lot of methods. Here are some results. So I'll take you through these. Let's see, am I doing this right? Yep, here's what we found. First, we have the effect of acclimation on maximum heart rate. We found that the maximum heart rate of our 4 degrees Celsius exposed snails was higher, suggesting that they have an increased capacity for oxygen delivery. Second, effect of exposure and peak temperature, our 4 degrees Celsius exposed snails also had their best, strongest performance at higher temperatures than those -1.8 Celsius exposed snails. Finally, the heart rate breakpoint. That breakpoint, again, for our 4 degrees Celsius snails was once again higher than the -1.8 treatment, suggesting that their hearts are better able to keep up with the metabolic demand, at higher temperatures, and also that they fail less quickly. So to put all these data together, and another way to visualize our findings, here is a compilation of all of the thermal performance curves for all 28 of our animals. Again, we have heart rate on the y-axis and temperature on the x-axis. Blue solid lines are the ones that are exposed to -1.8 Celsius, and the red dotted lines are the ones exposed to 4 degrees. So on average, we found that maximum heart rate increased by 2.6 beats per minute. Was 2.6 beats per minute higher in snails exposed to warmer waters. Warm exposed snails also had their best performance at temperatures that were on average 1.9 degrees Celsius higher, and their heart rate breakpoint was also higher by about 1.7 degrees. Now, putting all of these data together, what we can conclude is that, our 4 degrees Celsius exposed snails had better performance at higher temperatures than our ambient treatments on all three parameters that we are investigating. And we take this as strong evidence that these snails are able to acclimate to warmer waters in as little as 2 weeks, which is, again, is much, much shorter than what we thought that Antarctic ectotherms were capable of. So what this means is that these snails at least withstand current seasonal variations in temperature in McMurdo Sound, and perhaps most importantly, not all Antarctic invertebrates are going to take months to acclimate to warmer temperatures. Okay, with that conclusion slide, here's my thank you slide. I'd like to thank you, first of all, y'all, for your time and attention. Big thanks to the Antarctic diving support staff, Rob Robbins and Steve Rupp, Prairie Lab staff, and all members of the 2021 and 22 McMurdo community. Lastly, I'd like to thank actually the SCAR organizers and as well as our conveners, Fernanda, Fabiana, and Eugenia, for putting all of this together. Please feel free to email me if you have any questions. And lastly, if you enjoyed my talk, please tune in tomorrow at the same time, where my PI, Dr. Amy Moran, is going to tell you about how we recently cracked one of the biggest mysteries of sea spider reproduction. Okay, thank you very much for your attention.
Cool, thank you, Aaron.
Thank you.
Aaron was the last presentation, so we are now in the time of questions and answers. So we have some questions here. The first, the first was for Tamara, but Tamara is not among us right now. She has to leave because of problems with internet connection. So I would suggest to send her an email or something.
Yes, she wrote the email in the chat box,
yes.
so you can write her.
Yes. And about the amoeba was
The question was for Maywin Herliden. I don't know if it is correct. I will unmute her as a member to allow her to answer. Actually, I'm not able to do that. So we can try. She should be able to answer right now.
Yes.
Maywin, if the pronunciation is right, we have a question by Laura Zucconi who is asking if there are studies on amoeba diversity from continental Antarctic soils or wet environments.
Yes, I was looking for the name. Yes, Maywin, for Maywin.
Maywin, yes.
Can you hear me?
Yes.
Yes. Can you repeat the question, please?
There are some studies on amoeba diversity from continental Antarctic soils or wet environments.
Yes, there are. There are some studies, but I think the studies are from the 90s years, so not recently. But there are some studies in Antarctica.
Okay, thank you.
Thank you. More questions? I have. for Graham. Graham, do you know if there are some similar patterns for other groups of similar results that you have?
We only did the work with the one species. I know there's been some studies with sea urchins, Styrachinus, and they do show a somewhat similar, like they're very thermally or temperature sensitive, so those high Q10 values. But with ours, I guess what's really odd is the fact that in their natural temperature range, they're more sensitive than outside of it. I can't speak for, I guess, the rest of it, but there hasn't been, I guess, a whole lot of work done on that early development of these organisms.
Okay, thank you. And I have another question if nobody has a question. I have a question for Aaron. Aaron, is there a temperature when the mollusks die? Do you find this? Because I was just wondering if could be a relation between this acclimation with stress. If the mollusk is very stressed up to the point when it dies. Mm-hmm. Did you find something like this?
So I was quite surprised by these results, but none of our snails died at all for 2 weeks after we ran the temperature ramp on them. So again, that temperature ramp was stressing them from -1.8 all the way to 18 degrees Celsius. And I only stopped the experiment when all of the snails were exhibiting their flat line that has 0 heart rate. So all of these snails were pushed to 0 heart rate. And then after that, I took them, I put them back in their ambient temperature, and all of them recovered and survived for at least 2 weeks. So this 18 degrees Celsius is not enough to kill these snails. Of course, I will say that we exposed them for quite a short period. They were only held at each of those temperatures for 2 hours. So if we were to continually leave them at 18 degrees Celsius, they'll probably die. I would expect they would die. They would eventually die. Yeah.
Because there are some studies, quite old ones actually, on scallops, on the Adamusium, you know, that they die when it's something like 21 degrees. And just curious about the gastropods, but that
again, I think it depends on how long we want to leave the animals at a warm temperature. Mm-hmm.
Oh,
thank you.
Cool. Thank you. We still have time for more questions. 3 minutes, actually. Okay, I have a question for Maywin. Maywin, is that correct, the pronunciation? How is the
This is
correct?
Maywin.
Maywin, okay.
Yes.
So did you find in the literature same pattern you achieved in other areas, for other areas?
Not really, because the study of the state. Do you hear me?
Yes, yes.
Yes, I'm sorry because I don't have so much network. So, no, the study on testate amoebae on the past study are not how the study don't show these parameters from the testate amoebae. So there are some study on past climate, but they don't show the difference with the total organic carbon or other parameters. So this is a new kind of study for testate amoebae.
Okay, data, you have a difference in how to get the data, right?
Ah,
so
how?
you don't know what to expect, for instance, in the Arctic, because I think the comparison between Antarctic and the Arctic is very easy to think about. So do you know if there are some results on the Arctic or something?
Yes, there are also some results on Arctic, but I didn't do the comparison to already. So
okay, thank you. Okay, do you have more questions? Or we are done. I think we are done. Fabi, do you want to close the session?
Yes, I want to acknowledge all the speakers for their contribution, very interesting, everything, and also the attendees. I want to remind you that you can still make some question in the SCAR portal or writing to the authors, and I would also invite you to take a look to the posters that are in our session that are also very interesting. And I want to remind you that we will have other 2 sessions on the same topic tomorrow at 3 UTC and also at 7 UTC. So we want to invite you to follow these sessions tomorrow with other very interesting contributions. Do you want to add something? Fernanda?
No, it's fine. I appreciate the participation of all of you. Thank you for the discussion and for presenting your studies. All of them very, very interesting. Fabiana and Eugenia were very happy to see all the abstracts we received, because the session is very, very interesting and we really appreciate that. So we see you tomorrow. Or Eugenia, if you want to say something, please open your microphone.
Hi everyone. Do you can hear me? I have some issues with the
Yes. Perfect.
I want to thanks for your presentations, for all of your presentation. Thank you very much.
Okay. That's it, Fabi.
So see you tomorrow. I hope with every one of you. And goodbye.
Bye-bye.
Thank
See you
you.
tomorrow.
Bye.
Thank you.
Thanks.
Thank
See you
you.
tomorrow. Bye bye.
Okay. So
Sorry, do we have to stop the recording, or you can do that this? Sorry.
Hi, this is Manoj from Tech Team. Yeah, I think
Yes.
panelists also went over there. So if you want, I can stop the recording as well.
Okay, please.
okay.
Yeah, thank you.
Yes.
It's
Thank
been
you.
great
Thank you.
session.
Thank you.
Thank you.
Yeah,
In
we are more
time.
than happy to. Yes.
Good. Thank you. No,